Hello, everyone, and thank you for attending today's webinar. My name is Honrado Lopez, Associate Product Manager of Microfluidics at Fluidigm, based in South San Francisco, California, and will be today's webinar host and moderator. Our speaker today is Jess McDevitt from Infinity BiologiX, and she will be presenting how our company uses microfluidics in the development, validation, and implementation of custom genotyping panels in an industry environment. I'd like to spend a few minutes going over microfluidic solutions provided by Fluidigm, which the team at Infinity BiologiX utilizes in their workflow. Fluidigm offers several solutions for genomic analysis, which are driven by automation and microfluidics. Anchored by our core integrated fluidic circuit technology, which enables tens to thousands of nanoscale reactions per sample per run, Fluidigm offers tools for next-generation sequencing library preparation using the Juno system and real-time and endpoint PCR with the BiomarkHD, our flagship genomic system. We also provide fully kitted high-performance reagents in order to provide single supplier consistency and support, as well as custom primer design using our D3 assay design service. There are several key benefits that are enabled by our Fluidigm microfluidics-based platforms. Time-consuming steps like PCR reaction setup are automated through the use of IFCs and our platforms. Hands-on time is reduced, which maximizes lab efficiency and minimizes user pipetting errors. By moving from traditional microliter reaction volumes to nanoliter reactions, labs can significantly reduce costs of sample and reagent consumption. The BiomarkHD supports multiple genomic applications, from SNP genotyping, gene expression, and digital PCR, which alleviates the need for multiple single application instruments in your lab. In addition to being our NGS library preparation system for targeted amplicon sequencing and RNA-seq, Juno also doubles as a universal IFC controller that operates upstream of the BiomarkHD. And our IFC catalog also supports varying throughput needs for labs. IFCs can support anywhere from 144 to 9,216 individual reactions per run, all without the need of changing platforms or technologies. Our genomics platforms, whether for real-time PCR or targeted NGS library prep, all leverage the use of integrated fluidic circuits, or IFC. This yields both flexibility, scalability, and automation across many attributes, such as number of samples run or number of targets analyzed. And the movement and mixing of samples and reagents are controlled within the chambers of the chip itself. It's a hands-off process, saving many individual pipetting steps. Within the IFC, samples and assays are loaded into individual inlets in the carrier, and the IFC is placed in a Juno or standalone controller, which automates the reaction mixing step. Samples and assays are combined in individual nanoliter reaction chambers and undergo thermal cycling in the BiomarkHD. Each sample is partitioned and combined with each assay. In the example shown here of the 192.24 Dynamic Array IFC, 192 samples are loaded and are interrogated by up to 24 assays, yielding a total of 4,608 data points. Another key benefit to the IFC is that assays are not prefixed or pre-spotted, which allows panel content to be adjusted between IFC runs. This provides a simpler way to optimize or troubleshoot assay panels without committing to a typical minimum order quantity for prefixed panels. Utilizing microfluidics also saves labs time and money when running PCR studies. When comparing a traditional 96 well plate to our 96.96 dynamic array, the traditional method uses significantly more reagent and requires much more time in order to achieve the same level of data throughput as the IFC. Data throughput from a single IFC run is the equivalent of 96 well plates or 24 384 well plates. As I mentioned earlier, Fluidigm also provides custom assay design services for a variety of applications. Our D3 team can assist you with your targeted amplicon sequencing, gene expression, and SNP genotyping studies. Simply create a D3 account at fluidigm.com/D3 and define your targets of interest for your desired application. Our D3 team will then design and deliver the optimal primers for your study. And if at any point in the process you need assistance, you can reach out directly to our dedicated assay design specialists or connect with your local sales representative or field application specialist. Thank you very much for your attention, and now on to the main event. Today, we have the pleasure of hearing from Jess McDevitt, Associate Director of Development Projects at Infinity BioLogics, formerly known as RUCDR Infinite BioLogics. For the purposes of today's talk, you may also hear Infinity BioLogics referred to as IBX. Jess completed her undergraduate studies at Georgian Court University in 2008 and earned her master's in microbial biology from Rutgers University in 2020. Jess has been with IBX for over 10 years, working extensively with microfluidics. Critically, Jess was part of the team that developed the first saliva test for SARS-CoV-2 detection that received FDA emergency use authorization, and she played a key role in developing and implementing a workflow that enables IBX to have the capacity to perform 100,000 SARS-CoV-2 tests per day. In her presentation, Jess will share how IBX utilizes Fluidigm microfluidics to perform more than 200,000 genotyping reactions a day. And in collaboration with Fluidigm scientists, IBX has developed a variety of custom panels providing new methods to optimize throughput and provide high-quality data to clients. Jess, you have the floor.
Hey everybody, I hope everyone is well today. So I'm Jess McDevitt, and I'd like to welcome you to my talk on development, validation, and implementation of custom panels utilizing the Fluidigm workflow. Just a little bit about myself. As Conrado mentioned, I've been with Infinity BioLogics for about 13 years now, and I've spent the past decade working with Fluidigm technology, building new panels, oversight of lab processing, troubleshooting, and streamlining workflows. Over the years, I've developed multiple custom panels and protocols, and I've worked closely with scientists at Fluidigm to optimize protocols and find ways to efficiently meet client needs while delivering high-quality data that researchers can have confidence in. While this talk will focus on genotyping, IBX has also utilized microfluidics for gene expression and single-cell microfluidics. Just a quick overview of what we're going to talk about today. First, I'm going to give you a quick introduction to Infinity BioLogics. And then we're going to go over the importance of microfluidics while maintaining a biorepository. Then we're going to discuss the development and utilization of custom panels, how those custom panels are validated, and how those custom panels are then implemented. So first, I want to read a quote from one of our co-founders, Dr. Andy Brooks. Your biospecimens are as important to us as they are to you, and most importantly, to your patients and study participants. Our approach helps you preserve your precious assets for questions you need answered today, while at the same time, preserving your biological assets for discoveries in the future. IBX is a next-generation central lab supporting academia, government, and industry, and providing global sample collection, processing, storage, and analytical services integrated with scientific and technical support in both the research and clinical arenas. As a leader in biomaterials, IBX provides support to the development of diagnostics, therapeutics, and research in the genomics, precision, and regenerative medicine space. We have a patient-first philosophy here at IBX. We recognize that each sample coming through our laboratory is from a patient, and that the data we generate affects lives. We take that responsibility very seriously and are proud of our ability to accurately deliver data with confidence. In addition, as Conrado mentioned in the introduction, we are one of the largest COVID testing facilities in the country, with the ability to process 100,000 samples daily. We are most recently known for having the first EUA for saliva testing, which was followed by the first EUA for at-home collection. These EUAs changed the landscaping of COVID testing in a very difficult time in our nation. We continue to innovate in a COVID world to bring solutions to the complex problems associated with this pandemic. So first, I just want to give a quick little overview of different genotyping technologies. We have plate-based methods, which are better for lower throughput projects with just a few loci and a limited number of samples. These can be used for genotyping or gene expression and utilize probe-based qPCR. Microarrays, however, are ideal for large screenings and investigations into hundreds of thousands to up to 2.5 million SNPs. They are more cost-effective for extremely large data sets and high-scale throughput. You can run plates in sample numbers of 24, 96, or 384. There are multiple platforms available depending on your research needs. We can run genotyping, gene expression, and epigenetic panels. With targeted sequencing, a subset of genes or regions of the genome are isolated and sequenced, allowing researchers to focus their time, expense, and analysis on the specific region of interest. These panels often cover SNP quantities between what a microarray and microfluidics would cover. These are more ideal when you're looking at an entire gene or genes required, so a few thousand of SNPs. And the talk of the hour, microfluidics. Microfluidics is really great for targeted panels of greater than 24 SNPs, and when you're going to have high sample throughput and you require fast and accurate results. So why microfluidics? Why do we use them? Microfluidics offers a more cost-effective and high throughput alternative to plate-based methods. Using a 96 by 96 IFC as an example, which would be 96 assays by 96 samples, you can get 9,216 individual genotyping reactions in just over 4 hours with less than 30 minutes of hands-on tech time. In contrast, on plate-based qPCR, this process would require 24 384-well plates, approximately 5 days of processing time, and many hours of technician time. Microfluidics also reduces the potential for human error with less pipetting steps, easier sample mapping, and streamlined data analysis. With nanoliter reactions, expensive reagents are conserved, which significantly reduces your cost per reaction. As you can see in this image I've shown, the data quality is concordant with plate-based methods, making Fluidigm microfluidics an ideal choice for high throughput genotyping panels. I want to describe, before we get into the meat of the talk, some of the main things that you're going to be looking for when looking at genotyping data, especially with the Fluidigm platform. You're going to see a lot of these cluster plots throughout the talk, so I want to make sure that everyone is on the same page with what we're looking for. First of all, each of these dots represents a sample, and each of these graphs represents the assay. The green and red clusters are homozygous samples, whereas the blue cluster is a heterozygous call. So for example, these would be CC, these would be CT, and these would be TT. The quality of the clustering is also really important when you're calling your data. So what you really want is for your graph to look like this top graph here and not like this graph here. This bottom graph here, you can't have confidence in calling your data. I also want to call out the rocks images. So this image has the 9,216 reaction chambers, and you're able to see each one of these squares is the individual reaction chamber. You want your rocks image to look a lot like this one, with uniform loading across all assays and all samples. Sometimes, when looking at this image, you'll see blips across, and that usually indicates an issue in loading on the tech end, either a bubble or insufficient sample. If this occurs, it's not an issue with your assay or your sample, rather something that needs to be repeated. So I wanted to discuss IBX's use of Fluidigm microfluidics. One of the most important uses is our functional quality control panel, which will be discussed in detail in the next slide. Another is speed congenics, which is a test that we previously ran on another platform. To improve turnaround times and save on costs, we transferred it to the Fluidigm platform. This included working with Fluidigm to develop the 24 by 192 array so that 192 assays could be run with 24 samples. The original array ran 24 assays with 192 samples. We worked closely with Fluidigm to develop this new protocol, which allowed us to generate large amounts of data for small sample sets quickly and accurately. We also offer a variety of direct-to-consumer and wellness panels for our clients, as well as confirmatory qPCR testing. Confirmatory qPCR testing is often done when an area on the microarray has poor performance, yielding lower confidence in the genotype calls. In that case, we would use the gold standard process of qPCR on the Fluidigm Dynamic Array to confirm the correct genotypes at those loci. Functional quality control is an extremely important aspect of maintaining a biorepository. The SNP trace panel utilizes 96 SNPs, which are run on every sample that comes through the biorepository. This panel assigns a molecular barcode to every sample so that the integrity of sample identity can be maintained throughout every step of the life cycle that the sample has at IBX. Looking at this image here, each line across is a sample, and they're all from the same subject. When reviewing the data, you can see that the sample's genotypes match, which tells us that all of these daughter aliquots came from the same parent sample, and there were no swaps or issues with sample integrity along the way. Many biorepositories use STR testing for this purpose, but SNP trace has been shown to be more cost-effective with a high level of accuracy. In addition, the panel gives us a quality score on the sample. A combination of call rates, gender calls, and performance of critical SNPs gives an indication on how the sample will perform in downstream applications. Functional quality control is the first line of defense against sample misidentification. We do this by comparing the genotype gender against the site-reported gender. This can identify and often rectify sample swaps that happen before we even receive source samples. The panel can also be used to identify sample swaps within a family. For example, if the identity of the proband is switched with one of the parents at the collection site. The panel is also extremely useful in ensuring there are no issues with the sample throughout other steps of its life cycle here. When microarrays are run, their data is compared against the FCQC panel to ensure that no samples have been misidentified on manifest and no other human error that could affect mapping has occurred. For example, if there were plate swapping and plate inversion. Using this comparison, IBX is able to catch and correct potential errors before data is released to a researcher. This information can also be used to assure a client that the data that has been released is accurate should questions arise downstream of release. Any quality check we can put in place to catch or prevent human error is paramount in releasing accurate data. The functional quality control panel is extremely helpful in troubleshooting. For example, if samples are not performing well on a plate-based qPCR assay, we can go back to the FQC results to show that it is likely due to sample quality. Likewise, we can get ahead of this by seeing poor FQC results and either purifying or pre-amplifying the samples to get results clients need without any further delays. So now I want to go over what we're going to talk about when we talk about custom panels. The first step is identifying your SNPs of interest. In many cases, on our end, that's done by our clients, and we receive a SNP list from them. In a research or academic lab, that's something that you may identify from other literature or what you're investigating. The next step is selecting the appropriate array, your chemistry choice, designing your assays, and testing and optimization. So first I want to show you 3 different examples of arrays This array is a 48 by 48, so you can have 48 assays by 48 samples. This array is 192 by 24, and this is the one where you can have either 192 assays by 24 samples or 24 assays by 192 samples. And then this is the 96 by 96 panel, which does 96 samples by 96 assays. When selecting the appropriate array, you want to look at a few things. First is the number of assays in your panel and your expected sample throughput. If it is a large number of assays, you would want the 96 by 96 or 24 by 192. Which IFC you would select would depend on your expected sample throughput. If you're expecting large sample receipts in a short period of time, the 96 by 96 IFC would be best. However, if you have greater than 96 assays and are only expecting a small number of samples, the 24 by 192 chip is a better choice. You want to put all of these considerations into your calculations and into your cost analysis. Selecting the appropriate array for your project is an important first step in moving forward with the development of your assay. So I want to discuss the different chemistry choices. Another chemistry that can be used with Fluidigm is a competitor chemistry, so I don't want to go into the name, but it is more specific is one of the pros. It has a fast chemistry available so that you can get your reactions in just over an hour, and it has a streamlined workflow. The cons of this chemistry is that it is expensive, and the custom design tool is less user-friendly. The Fluidigm SNP type chemistry has some definite pros. The user-friendly design tool is great and easy to work with, and it has comparable performance to other chemistries. It is much more cost-effective, and I cannot say enough good things about the technical support that I received from Fluidigm, especially when dealing with a difficult panel. The con can be that some loci can be a bit more challenging as it's less specific, and the workflow can take a little bit longer, but you're still getting your reactions in a comparable amount of time. So now I want to go over your workflow for development and validation. Your first step is assay development, followed by troubleshooting, assay optimization, getting your working assay, assay validation, control selection, study design, and then validation execution. So assay development, you can have custom or off-the-shelf assays. Off-the-shelf assays usually work as soon as you get them in the door and you really don't have many issues with them. Custom assays can be a little bit more challenging and may require some optimization to get them to work. Sometimes you may have assays that are unable to be manufactured due to difficulty at the locus. And in that case, you may need to go to a surrogate SNP that gives you similar results. You may have to attempt multiple designs. I've had times where we attempted 5 different designs before we got one that worked, but it was worth it. And then you want to look at your initial assay results. So this is an example from a recent panel, and this was an assay on demand that performed pretty much perfectly on its first go. This one, however, was a custom assay, and we tried 3 different designs and could not get it to work, and we ended up going with a surrogate SNP that gave us similar results. During the troubleshooting process, you can have issues with poor clustering, where you can't really make calls because the clusters are not only spread out, but really close together. Low amplification, which while this has nice tight clusters, if I have samples with lower yield, I may have some issues getting accurate calls, or no amplification at all. When you have no amplification at all, the first thing you want to do is look at that rocks image that I showed you earlier and make sure you have accurate loading throughout the chip. If you do, then it is likely due to assay design and not a loading or technical error. So once you have something like this, you want to go forward to optimizing your assays and seeing how you can get them to perform better. One of the first things I look at is the DNA input. So for this particular assay that we were optimizing recently, I tried 60 nanograms per microliter to see how that worked. And while it sounds counterintuitive, I also tried 30 nanograms per microliter to lower the amount that we had. Sometimes with certain assay types, the lower input works better. For example, with saliva, you sometimes have better results when you dilute them more than with, say, a high quality sample source like whole blood. So we still don't have results that I'm really happy with here. So we went on to the next step of optimization for this panel, which was preamplification. Preamplification is a PCR step that takes place prior to chip loading. What you do is pool all of your assays into one reaction with each individual sample for specific target amplification. After this step is complete, you move forward with the chip loading process using your preamplified material. This increases the number of amplicons for each assay in your panel. The increased quantity of amplicons can result in cleaner, tighter clusters, and more accurate data. In this case, we tried it with 60 nanograms per microliter and 30 nanograms per microliter. So, as we've discussed, we want those nice tight clusters with differentiation, and the preamp with 30 nanograms per microliter worked. Now, I want to call something out that's really important, is that anything you try when optimizing, you need to make sure that all of the assays in your panel can support the changes you're making. So if you have one assay on your panel that's performing really well and has great amplification, and then one that's performing with low amplification, changing your DNA input could affect one assay or the other. So you may have to play with a lot of these changes before you get something that has a great working panel across all of the assays. For this, we went with 30 nanograms per microliter input with Preamp. Now at this stage, our development is complete and we can move on to validation. So the steps of assay validation are control and sample selection, study design, documentation, and then you're going to look at accuracy, analytical sensitivity, analytical specificity, and precision. For control and sample selection, the first thing you want to look at is your controls. And your controls are going to be samples with known genotypes, whether they're reference samples or something that you have in your repository. Ideally, you want to cover all possible genotypes across all assays. This can be difficult if you have a large number of assays and a small number of sample spots on your chip. It's also important that any controls you use in your validation are used in your live runs. In our most recently developed assay, we were only using 7 SNPs in duplicate, which meant we could use controls to cover all genotypes, which gives us increased confidence in the calls you're making, especially if one of the calls has a lower frequency in the population. And then you want your test samples. You need a minimum of 20 samples, and these samples should go through end-to-end lab processing. During this process, you want to be validating everything from your sample collection device, your extraction process, your DNA normalization, and your genotyping. You also want to test all sources that you're expecting. If you're just expecting saliva, then you just need 20 saliva samples to do your end-to-end processing. But if you're also expecting buccal swabs and whole blood, you'll need to test them all to make sure that they're all working and that you can validate them all for use in your laboratory. Study design. So something that's really important in study design is documentation. You want to create a plan that has the procedures you're going to follow, which means lists of your SOPs, descriptions of your methods, and you want to clearly lay out your acceptance criteria. For genotyping, your acceptance criteria should include accuracy. Accuracy is the concordance of your samples to expected results. So for example, if you have reference samples with what results you're expecting, you should make sure that all of the genotypes are concordant. You need to also look at your analytical sensitivity. and this is the likelihood that the assay will detect the targeted sequence variant when the variant is present. For example, a true positive result or a true variant result. The calculation for this is the number of true positives divided by the number of true positives plus false negatives. You then need to look at analytical specificity. This shows that the assay will not detect the targeted variant when the targeted variant is not present. For example, a true negative result or a true wild type result. The calculation for this is the number of true negatives divided by the number of true negatives plus false positives. Precision has three parts: repeatability, reproducibility, and intra-lab reproducibility. Repeatability is the samples performing in one array. So what you want to do for repeatability is run your samples in triplicate on one array and analyze their concordance. For reproducibility, you want to run your samples on 3 different days, preferably by 3 different techs on 3 different instruments. I recognize that this is not always possible in the lab based on the number of instruments you have, but this is what's ideal. Intra-lab reproducibility is when you randomly select several samples and send to an external laboratory to check and make sure that the results they're getting match the results that you're getting. Your acceptance criteria for these steps is 95%. So validation execution. You need to follow your SOPs and your validation plan. Any deviations you need to document with explanations. For example, if you ran out of one of your samples and there was not sufficient quantity to continue, and you moved a different sample into the validation, you need to explain that. And then you need to look at if your acceptance criteria were met. Did your validation pass and can you move forward, or do you need to go back to the drawing board? But once our validation passes and everybody is happy, we move on to implementation. Implementation involves a few things. You need to look at your equipment needs, inventory management, SOPs, training, data handling and report generation, and end-to-end user testing. When looking at your equipment needs, you want to analyze your in-house infrastructure with your available techs and the number of shifts. I really want to emphasize that in your equipment needs, redundancy is important, especially if you have tight turnaround times. So for example, if you only have one endpoint reader or one biomark and it goes down, then you can't do anything until it's able to be repaired, which can significantly push back your turnaround time. Inventory management is something that's really important in a post-COVID world. So you want to look at your expected needs and sample throughput and start setting up standing orders if possible. And when I say standing orders, I don't just mean standing orders for your chips and the reagents from Fluidigm. I mean standing orders for all consumables that you need. Everything from reservoirs, foil seals, plates, pipette tips. These all need to be accounted for as there have been a lot of supply issues in the last year. So for SOPs and training, your SOPs should all be written before you validate, and they should stay as per the validation. And your SOPs should include your QC metrics, which should be identified during the development phase, and a troubleshooting guide for the lab so they know how to fix problems and when they need to escalate up to management. You also need training documentation. You need to document the training for all steps of your SOPs to ensure that you have technicians trained for each piece of the process and ready to go when you go live. The next thing that is really important is end-to-end user testing. It is important to test and document every step of the process with dummy samples before going live. You want to start with the receipt and registration of your samples into your LIMS system. Then the samples are going to be transferred to the lab. What mechanism are you going to use to ensure the lab performs the appropriate process on the samples as they move through? Does this mechanism work? How is it tracked in your LIMS system? You want to test the extraction process. Are all the files you need for downstream processing properly in your LIMS system? Are you able to use these files for your DNA normalization And is your DNA normalization going appropriately? Then move on to your genotyping. How are you attributing sample data in a way that decreases the potential for human error? It is ideal if your system either creates your plate map for you, or if you use a position-based plate map every time, and then map the samples downstream in the data analysis and bioinformatics software. You also want to look at your data analysis. Is this process working? Are the technicians able to perform data analysis properly? Can your technicians export this data and transfer it over to bioinformatics? Can bioinformatics then take your export Fluidigm files, map them to the samples and genotype results appropriately, and put them into a format needed to easily read the data and transfer to the client? Every single step of this process is equally important. Even a little hiccup anywhere along the way can halt your process or introduce human intervention, which can then cause error. This can then affect your data release and your ability to meet timelines. So some of the acknowledgements I wanted to give were to Dr. Shriznaha, Robin Grimwood, Russ Hager, and in loving memory of Dr. Andy Brooks, our co-founder.
So we have a few questions here for you, Jess. The first one, what is the typical turnaround time to develop and optimize an assay panel at IBX?
About 6 to 8 weeks.
Okay, great. And that goes through the entire workflow from development and like troubleshooting and also identifying your controls?
So those are identifying your controls and everything are things you can begin during the development process, as I usually use some of the controls during development. However, development can take a little longer. It just depends on if your assays work the first time, if you have to order additional assays, because that can push you back depending on how long it takes to receive your new custom assays.
Okay, great. Okay, we got one more here. So you referred to your functional quality control test, I think using the RUID panel. So what are the steps taken if a sample fails that quality control test?
Right, so if a sample fails your quality control test, it depends on why it is failing. So if it's failing because of a profile match or profile inconsistency, you want to go and look back at the sample's life cycle and see if there's somewhere where it was potentially swapped, or if there's somewhere where it was attributed incorrectly at the site. And with profile matches, sometimes it could be that it's the same participant for different studies, but they get different subject codes. So it may look like a profile match, but it's actually just the same person who's participating in multiple studies. If it's failing because of quality, it really depends on the level at which it's failing. If your sample fails outright and you have almost no calls on, less than 50% call rates, it's really likely due to the quality of your sample, and it's not going to do well on downstream applications. So the sample may need to be either recollected, re-extracted, or purified in a DNA pure process.
Okay, great. So I'm seeing no further questions this time. I think we're going to wrap up today's talk, but if there are any questions that come up, after we close, feel free to send them over to Anita Smith, who is on our regional marketing team. And that email address is anita.smith@fluidigm.com. And she'll forward it to, to either your local sales representative or a field application specialist, and we'll get an answer right to you. I want to thank Jess for the great talk this morning. And yeah, have a great day, everyone.